Hi.
Hi, 안녕하세요.
Hi.
Ebola virus, known as a hemorrhagic fever, transmitted to people from wild animals and spread in human population. How does it spread? Human to human, blood of body fluid, body of a person who died from Ebola.
So there are several symptoms can be seen on people who diagnosed with Ebola virus through the incubation period. So the incubation period, that is the time interval from infection with the virus to onset of symptom is from 2 to 21 days. So some of the symptoms are fever, fatigue, muscle pain, headache, sore throat, vomiting, diarrhea and rash.
First possible way that we can do is by doing isolation and synthetic production of human monoclonal antibodies. This process allows a production of antibody by phage display only. To come up with Ebola vaccine that called RVSV-ZEVOV, we use RT-PCR, which is reverse transcription polymers, which combine protein from Ebola virus and DNA from VSV, Versicolor stomatitis virus, to form vaccine. This VSV then been cloned into expression vectors. So why do we use VSV? This is because VSV-G protein bind with numerous cell types and actually attack animals while cause flu-like illness to humans. This strategy is similar to the approach used to eradicate smallpox. The volunteer very rat protector. The scientists will take a blood sample from each volunteer before and after they get a vaccine to see the progress. We also can do ring vaccination, where individuals at risk of infection will give a vaccine.
So I will explain to you how VSV vaccine will work in our human body. So before I will explain to you how VSV vaccine will work in human body, we have to know the structure of Ebola virus first. So as we can see on this paper, the structure of Ebola virus is divided into two parts, which is payload and delivery systems. So the payload consists of RNA. RNA is a genetic code that allows virus to take over your cell and turn it into a virus factory. And then this RNA, which is the code that will deliver it into cell by delivery system, that consists of membrane and glycoprotein, as you can see in the paper. So how does this vaccine work on Ebola virus? So VSV vaccine is a virus that is not harmful. It will replace its delivery system of its glycoprotein with glycoprotein of Ebola virus, as you can see in this the arrow of orange, that is the glycoprotein is replaced into VSV vaccine. So the result, it will result in VSV with glycoprotein from Ebola, and Ebola, it has Ebola delivery system, but its payload is actually not harmful genetic material inside our body.
So, as we know, Ebola virus directly attacks immune system. As a scientist who is trying to develop an antiviral drug, I would like to explain one of the possible ways to obtain the antiviral drug. So, the antiviral drug can be produced by ZMapp drug. A ZMapp drug is a mix of 3 monoclonal antibodies which bind to the Ebola virus. The virus will then not be able to enter the host cell, which needs to multiply itself. The body's immune system then able to recognize the virus and eliminate it. The monoclonal antibodies are produced by injecting a mice with a protein of Ebola virus. The mice produce antibodies which then bind the Ebola protein. The mice antibody needs to be modified in a way that they resemble human antibodies to avoid any immune reaction against the antibody. This process is called humanization. Thank you.
Right now I want to explain to you guys about the humanization of immune cell. Genes inside this cloned immune cell were humanized by replacing mouse portion with human DNA. The new genes with Ebola fighting antibodies are fitted into a virus that usually infects tobacco plants. This is because tobacco plant is a good medium to grow antibodies because they grow fast. Plants are less likely to transmit the virus compared to animals. ZMapp uses 3 antibodies to work together to fight the Ebola virus, 2 of which prevent the Ebola virus from binding itself to the host cells. The part of the ZMapp will target the infected cells to be eliminated by the immune system by the process phagocytosis. Thank you.
So, in conclusion, it can be summarized that VSV Evolve is safe and efficacious vaccine for use in humans. While this vaccine is still not licensed, several clinical trials are ongoing to provide further data to expand the efficacy and safety profile of the VSV Evolve vaccine. Although several other Evolve vaccine platforms have been developed and progressed to clinical trials, there are several unique advantages of VSV Evolve. The advantage of VSV Evolve is the rapid protection after vaccination and its potential post-exposure efficacy. The post-exposure treatment properties of the VSV Evolve vaccine have been used on several occasions in laboratory and healthcare workers exposed to and potentially infected with Evolve. The use of the VSV Evolve vaccine in thousands of people during phase 2/3 clinical trial also show that it's feasible to manufacture large-scale vaccine batch for deployment in outbreak situations.
Ebola outbreak in West Africa is the most severe in terms of death since 1976. Started in Guinea and spread to Liberia, Sierra Leone and DR Congo. So far, no Ebola treatment exists. Tests in monkey gave promising results when used ZMapp. 2 researchers were infected with Ebola during work in Liberia, and they were treated with ZMapp, and they were discharged from the hospital after a few weeks and showed positive results.